Okay, thank you very much for coming. I'm going to speak about comparing residue-coevolution network across homologous protein families. So, as most of us know, covariation of two columns in an MSA is an approach to residue coevolution. And though the calculation of the covariation score is between pair of position, that form a network. So our question was, can we compare covariation networks? And the answer is yes. In the same way we compare protein-protein interaction network or gene expression network, we can compare coevolution networks. We know that coevolution networks give us information about, for example, contacts, but we have other pairs that also coevolve, but they are not in contact. Are those false positives or noise? So we are going to compare covariation network between homologous proteins. For doing that, we use entire data set. That was a conserved domain database where each domain family is built based on sequence identity, structure similarity, and a lot of manual curation. That includes function assignment, ontology, and expert knowledge. So a conserved domain hierarchy has all the sequences which are all homologs. In the scheme, we have represented the dendrogram on the right side, and all those sequences are the ones inside the hierarchy. It is not easy to divide that in conserved domain families. Is made on sequence similarity, structure similarity, function annotation, residue annotation, but it's not an easy task, and ideally, inside each conserved domain family, we will have orthologs, and between families, they might be paralogs. So though the domain families are constructed taking into account protein and structure similarity, these two features are not evenly represented in the tree topology. That means that two conserved domains located in one branch of the alignment may have more sequence and structure identity than other two conserved domains which are located in another branch of the alignment, despite being at the same level of the hierarchy, at the same level of the tree topology. So for that reason, we define a set of classes based on the tree topology. So if we compare two domains located in different branches of the hierarchy and sharing no common ancestors apart from the root, we will say that these domains are poorly related. Instead, if we compare two domains located in different sub-branches of the hierarchy but share a common ancestor, a common ancestor apart from the root, we will say that these two conserved domains are moderately related. And at last, if we compare two conserved domains which have the same common ancestor and are at the lips of the tree topology, we will say that these two domains are highly related. So, as a description of our data set, we can see the distribution of the structure similarity and the sequence similarity between conserved domains, and with the red line we will see the highly related domains comparison. The green line shows the moderately related, and the blue line the poorly related. In this plot, it is worth noting that for example, the sequence identity, even for the highly related, it is very low. All the values are below 40% identity. Also, the structure similarity is worth having in mind that a TM score, which is a measure that speaks about the structure similarity between two proteins, two structures, if we have a score that is above 0.5, we can say that these proteins have the same fold. And we can see here that we have a lot of structures, comparison between structures that have a TM score below that threshold. So we have the hierarchy. For each of the concerned domains, we have a multiple sequence alignment, an MSA. From each multiple sequence alignment, we construct the coevolutionary network. And in that point, we made two flavors, one based on the mutual information in our own mutual information algorithm, which is a modification of the raw mutual information. And we also try with two methods which are based on direct coupling analysis and are called to be direct information methods. So how will we compare these two networks? Okay, we will use the graph edit distance at the beginning. And this is the graph edit distance is defined as the number of operations you have to do to transform one network into the other. That means how many edges you have to add or remove. This score goes from 0 to 1, meaning 1 that the two networks are equal, and at the other one round. If you have a 1, it means that the two networks are totally different, and if the score is near to 0 or 0, that means that the networks are equal. So, as every node in our network corresponds to one column in the alignment, and structural alignment will allow us to align the MSA, and so in such a way we can assign one-to-one the nodes of one network into the nodes in the other network. So at the right side, you will see the distribution of the graph edit distances. We can see that the highly related have the lowest graph edit distances. That means the best scores. And here we have just for comparison the sequence similarity and the structure similarity. So we calculated the area and the ROC curve for the different methods to distinguish between those classes. And we can see that for the different comparison of pair of classes, higher versus poor, moderately versus poor, and higher versus moderately related, that the TM score, this score that speaks about the structure similarity, or the mean sequence identity score, gives quite good performance for distinguishing those classes. But though the mutual information is not the best one, it gives quite a good predicted performance. So at the first time, we wanted to know if that is because of the similar in the structure. So here we have the distribution of the graph edit distance of the mutual information network. So we decomposed it and wanted to see and what in two subnetworks, which is the graph edit distance of those residues that co-evolve and are in contact. So it was higher, the prediction of the performance of this score to distinguish between classes was higher than the general one, but surprisingly, if we calculate that score between the residues that co-evolve and are not in contact, is even higher. Here we have as a comparison that the two measures of structure similarity and the sequence similarity gives much better performance for distinguishing those classes. So till now, we've used alignment-based comparison. That was useful to calculate the graph edit distance, but it incorporates some information, more information, which is the structure, that we might not have for comparing those networks. So we wanted to do alignment-free comparisons. In such methods, you don't need the structure, you don't need to align the alignments, so you will compare alignment-free the networks. That was based with the graphlet network decomposition that was introduced by Prue, and we will see 3 of the many distances that algorithm can give you. We are going to analyze the graphlet correlation distance and the relative graphlet frequency and the spectral distance. With that, we will calculate the area and the ROC curve to distinguish between those classes. So here we can see the distribution of the different methods. In the first line, we will see the highly versus moderately related families with the different distances. We are almost finishing. In the second line, highly versus poor, and in the third, moderately versus poor. In that slide, there are two things which are important to see. As a comparison, we made a network with the contacts in a structure, and we can see that for most of the distances, the contact network gives much worse results than the covariating networks. That is the first thing we have to take into account. And the other thing is that the mutual information network most of the times give higher results than the other direct information networks. With a star, we show the ones that perform the best for each comparison. So as our conclusions, we want to say we use covariation network to compare protein families. Networks of co-evolving pairs not in contact perform better than the full covariation networks. Related families have lower graphite distance and graphlet-based distances, and also graphlet signatures could be informative when no structure is available. The future perspectives are to explore node-based feature for predicting functional residues, infer function of residues, that is because residues in similar coevolution network may have the same function, to help protein annotation, and to see its suitability for protein classification. That's all. That work was done mostly by Dr. Franco Simonetti, who cannot be here, and now we incorporated another student, Guido Salustri, and those are all the members of the bioinformatics group in the Lelwar Institute in Buenos Aires, Argentina. So thank you very much for your attention. That's it.
Thank you for the presentation. I have one question and one comment. So comment first, maybe. So your mutual information-based networks perform better. Can it be probably due to the fact that they are simply denser? So there are just more edges in them.
They are not denser because we took a top 5% scores. We have an hypothesis why it performs better. With the direct information methods, you know that A and B coevolve and B and C coevolve. That with the mutual information algorithm, you have also that A and C coevolve. And that is a transitory relationship. As the direct information methods remove that. That is perfectly good to find contacts, but we have the belief that though not in contact for making a specific function, residues A, B, and C has to coevolve. I think that's it, is our hypothesis.
Right, and the question was actually, did you, so when you were calculating the correlations, did you control this for just conservation of positions? So how did you deal with, did you include the conserved positions into these networks, or did you exclude them? And did you then in any way compare your networks with just normal conservation signatures of your families?
No, we didn't remove the conserved columns. Take into account that conserved columns do not covariate because do not variate. Though we know from our experience that if it is quite conserved and not fully conserved, they might have a relationship, a covariating score. But as we are taking the highest covariation score, the top 5% of the scores, we think that we are not having the most conserved residues.
Hi, I had one question regarding the diversity of sequences in your families. I just wanted to know whether all of your families had enough diversity of sequences to calculate mutual.
All of my family have?
Like all the protein families that you analyze, like the multiple sequence alignment that you analyze, were informative enough to identify sites by mutual evolution? If not, how much percentage of your database would have families which cannot be used for this analysis?
Okay, that's our database. It's here. We control for sequence identity, for diversity. We use families that have 50 or more sequences. The ones that have lesser than that number, we didn't use it. So, and we also control for the diversity. We use sequences that are less than 62% identicals. So we have at least 50 sequences and less than 62% identical. And in such a way, we analyze 54 hierarchies that has 300 conserved domains and around 2,000 comparisons.
Thank you. Yeah, thanks.
So I'm struggling with jet lag, so this might be a terrible question, but when I
Me
saw
too.
you,
I'm struggling with loss of sound, of dreaming.
so when I look at this, I wonder what you think about using related things of this system to think about positive selection. So when you have your different kind of related families and you see different graphs and things, does this help point out clusters of positions that are under positive selection and maybe help us get beyond a lot of positive selection models, which are sometimes really focused on single columns at a time? But again, this might have misinterpreted some of this.
Okay, we have no idea about positive or negative selection, but as I told the other lady, we think we are not seeing the most conserved columns. I don't know if it answered your question.